Good afternoon, distinguished guests, ladies and gentlemen. Welcome to the Innovation in Agriculture and Food panel of the Science for Life Symposium today. I am Tai Xiang. It is my honor to be with you. Thank you so much for your esteemed presence. Thank you. Distinguished guests, ladies and gentlemen, the VinFuture Prize is a prestigious global science and technology award, proudly originating from Vietnam. And the topic we'll explore in today's panel are essential for advancing global scientific progress and have profound implications for human life. As we may know, agriculture is undergoing a major transformation driven by the urgent need to feed a growing population and protect our planet. Innovations in technology, data, and sustainable practices are opening exciting opportunities to increase productivity, optimize resources, and build a resilient agricultural system. Advances in genetic research are enabling the development of improved crop varieties. These breakthroughs in crop genetics and breeding are enhancing the food security, supporting sustainable farming practices, and helping to ensure a stable supply of essential crops. And together, these innovations are not only shaping the future of farming, but also the future of humanity. And to come with us today, it is the presence of many representatives from science and technology. It is our honor to introduce to you the presence of Professor Sir Richard Henry Friend, the chair of VinFuture Prize Council, University of Cambridge, United Kingdom. Welcome, sir. It is our honor to welcome Dr. Lê Thái Hà, Managing Director of VinFuture Foundation. And last but not least, it is our honor to introduce to you Professor Ingolf Steffan-Dewenter, Chair of today's symposium, member of the VinFuture Prize Pre-Screening Committee, University of Würzburg, Germany. Thank you so much for coming with us. Ladies and gentlemen, and now could you please kindly direct your attention to the main screen to introduce the symposium today's topic and also get acquainted with our distinguished chair and distinguished speakers through a brief video. So video, please.
Global agriculture is entering a transformative era as the combined impacts of climate change, resource depletion, and rapid population growth exert unprecedented pressure on food systems worldwide. According to the United Nations, the world's population is projected to reach 9.7 billion by 2050, driving a sharp increase in food demand. Meanwhile, farmland and water resources are rapidly shrinking due to climate change, urban growth, and overuse. More frequent and severe weather events, such as droughts, floods, and storms, have disrupted farming and global supply chains, causing losses of over $3.8 trillion in the past 3 decades. The future of agriculture will no longer rely on expanding farmland, but on innovation and higher productivity. Technology has become the driving force behind a new era of farming, one that is more efficient, resilient, and sustainable, ensuring food security for future generations. To promote scientific exchange and build strategic pathways towards sustainable agricultural systems, the VinFuture Foundation is honored to present the Symposium, Innovation in Agriculture and Food. The symposium will be chaired by Professor Ingolf Steffan-Dewenter, Chair of the Department of Animal Ecology and Tropical Biology at the University of Würzburg, Germany, member of the VinFuture Prize Pre-Screening Committee. He is a pioneering researcher in studying the impacts of global environmental change on biodiversity and the essential services provided by ecosystems. The symposium features distinguished international scientists in the field of agriculture, biology, and environment, including Professor Ermias Kebreab, University of California, Davis, USA, member of the VinFuture Prize Pre-Screening Committee. He was honored with the Top-AgriFood Prize by World Food Prize Foundation, 2025. Professor Raphaël Mercier, Director of the Department of Chromosome Biology at the Max Planck Institute for Plant Breeding Research, Germany, is a leading expert on the mechanisms of meiosis and genetic recombination in plants. Dr. Nadia Radsman from the Sainsbury Laboratory, Cambridge University, UK, She is an active steering committee member of Cambridge Global Food Security and a college research associate at the King's College Entrepreneurship Lab. Professor Pamela Christine Ronald, University of California, Davis, USA, member of the VinFuture Prize Council, recipient of the Wolf Prize in Agriculture, 2022, and the VinFuture Special Prize, 2022. She was named one of the world's 100 most influential people in biotechnology by Scientific American, 2015. In the spirit of rising and thriving, the theme of the 2025 VinFuture SciTech Week, the symposium will serve as a dynamic forum where science and technology inspire innovative solutions and lay the foundation for a green, safe, and sustainable future.
Thank you so much for your attention on our introductory video about the symposium's today's topic. Ladies and gentlemen, and now it is my honor to welcome to the stage Professor Ingolf Steffan-Dewenter, Department of Animal Ecology and Tropical Biology at the University of Würzburg, Germany, and he's also the chair of today's symposium to deliver the introductory presentation on our topic, Innovation in Agriculture and Food. And now it's your time. Thank you.
Yeah, thank you very much. Distinguished guests, dear colleagues, a warm welcome to the afternoon symposium on food and agriculture. And I would like to give a brief presentation to put the topic in a broader context, in a broader framing. Talking about food and agriculture. So, as we heard in the introductory video, food demand or demand for food will increase by 100% in 2050. We, on the other hand, observe that there's stagnating yields or even declines in yields for major crops in several production regions. And on the other hand, we have data that indicates that up to 50% of yields are lost after harvest, so that we could have a much higher food production or food availability if we wouldn't waste a significant proportion of food. On the other hand, agriculture has severe environmental impacts. So it's not just that climate has significant impacts on agriculture, it's also the other way around, that agriculture is contributing to greenhouse gas emissions, that we have pollution of water, soil, and the environment. I can't see the presentation here on the screen. That would be quite useful. We have this expanding agriculture, the destruction of natural habitats, ongoing biodiversity loss, and we also have soil degradation and soil erosion in agriculture. So we have lots of challenges, and the question is, how can we close the yield gap? How can we increase food production on the area that is under agriculture management? And we can assume that, or we observe that with current technologies, with current production systems, we have a yield gap. Currently, it's estimated that up to 30% of yields are destroyed by pathogens, destroyed by predators, or are lower than they could be due to lack of pollination services, for example. And this figure here illustrates you that we have different options, that we can increase yields by promoting ecosystem services, by improving soil quality, by enhancing pest control, natural pest control, and by promoting pollination of crops. And this way, we might be able to close yield gaps based on ecosystem services provisioning. So just to give you one key example that biodiversity matters for these ecosystem services. So you see here an example of a global synthesis work analyzing the relationship between the diversity of pollinators or the diversity of natural antagonists and how this contributes to the pollination services and to the pest control services in major crops across the globe. And these patterns underpin that biodiversity is part of the solution, that maintaining biodiversity in crop production systems can contribute to higher yields and to better natural pest control, and can replace part of the external inputs we rely currently in agriculture. So what kind of solutions from this perspective of agro-ecosystem could be achieved? So there are several transformative pathways for sustainable and resilient food production systems. And these can be, for example, at the field scale, mixed cropping systems, intercropping, organic cropping systems, or regenerative agriculture, as you see here, which is implemented in the European Mediterranean regions to counteract negative impacts of extreme drought and heat events. Or it can be agroforestry. And we also know nowadays from many studies that at the landscape scale we can promote crop yields by increasing crop diversity, by establishing green infrastructure, or by managing what we call functional biodiversity. So there are technological solutions that are building, that are based on a better ecosystem understanding. And in our session today, we will learn a lot about other technological contributions to food security and environmental sustainability. So we will learn about climate resilient crop varieties, about breeding technologies to have new crop varieties, also about climate smart livestock innovations and innovations to improve plant microbial soil interactions. But my important aim is here to demonstrates that we need to integrate a diversity of solutions to really have a translational change for ecosystem, for food security and environmental sustainable for future agriculture production systems. So we will have these 4 presentations and then a panel discussion and some closing summary and remarks. And I'm very much looking forward to the presentations for the next 40 minutes. And thank you for your attention.
Thank you so much, Professor Ingolf Steffan-Dewenter, for your amazing introduction. And we thoroughly understand about the symposium today's topic and the next part that we could see and we could meet our distinguished speakers. Ladies and gentlemen, and now it is time for us to introduce to you Professor Pamela Christine Ronald, University of California, Davis, USA, and welcome Professor for the very first presentation on From Genomes to Grains, Re-engineering Rice for a Changing Climate. Welcome Professor to our stage.
Thank you very much. Thanks to the VinFuture Foundation for putting on this great event, and thank you to our chair Ingolf for putting together this session. So today I wanted to tell you about a little bit of new work in my lab, and of course it comes back to this question that has already been articulated, at how do we feed 10 billion people? And of course there's no one way to do it, and we're going to need lots of tools. And one key aspect is we don't want to use more land and water, and we also want to reduce inputs such as harmful chemicals. But today I'm going to talk about a point that Ingolf touched on, is agriculture contributes quite a bit of greenhouse gas emissions. And in particular, I study rice, which is a large emitter of methane. So rice production is responsible for 12% of total methane emissions in the world. So our team decided to see if we could use a genetic approach to alter and reduce methane emissions. So first, let's talk a little bit about how methane's produced. So here you have a rice plant, and it's growing in an anaerobic environment, which is very favorable for methanogens that are producing the methane. And rice, like all plants, uses carbon dioxide through photosynthesis. to create different types of carbon compounds, about 20% of the carbon dioxide that's fixed in plants actually ends up underground. And part of that are secreted molecules, very diverse kinds of molecules that will affect the rhizosphere, which is the part of the soil underneath near the plant. And when you have changing compounds, this is going to affect the many microbes that we see in the ground. So we have a very diverse number of microbes, and these microbes, some of them, the methanogens, produce methane. So our goal is to see if we can slightly perturb this system so we can produce less methane. And we are rice geneticists, so we started with a genetic approach. This is a slide. I was trying to make it go. It shows you a conventional rice seedlings, as well as some rice seedlings that are expressing a small peptide hormone that we and others have been discovering. I think the movie, movies are so fun. Okay, well, what that was supposed to show you is that the PSY1 rice has roots that grow really fast and they grow longer. So the change of a single gene can affect the roots. Well, if we're affecting the roots, how is that affecting the microbes? So we carried out an experiment using agronomic soil from our local rice fields. So I live in Davis, California, where we grow rice. And we then potted up the soil, put the plants in the greenhouse, and then we monitored methane emissions over the lifetime of the plant. And we found cumulative methane flux is shown here on the y-axis, and on the bottom shows you the different plant types. So the rice control has quite a huge methane flux, but our two PSY lines containing this peptide hormone, two completely independently generated lines, both have about a 40% reduction in methane. And soil alone does not emit methane. So that gives you an idea that it's the rhizosphere, it's the microbes that are near the roots of the plants that are producing this methane. So our next step was to analyze the soil communities. We're lucky to work with collaborators like Jill Banfield at UC Berkeley to carry out metagenomic and metatranscriptomics analysis of the microbes. And we found that the transcription of one key gene, microbial gene, which is called MCRA, which is the last step in the production of methane emissions, is lower in our PSY plants. So that can partially explain this difference that we're seeing. So we are continuing to analyze our soil communities, and we want to understand which secretions, we call them exudates, so what molecules are feeding these microbes. And then we are carrying out synthetic biology approaches to change the secretions produced by the rice plants to alter that community and alter those soil microbe associations. We also have a fairly new tool of rice genome editing that many of you, I'm sure, know about, where we can make precise changes in the rice genome, altering the secretions from the root, then affecting the soil microbe interactions. So we're going from plant genetics to beneficial plant microbe interactions to reduce methane. We're also taking a look at carbon dioxide drawdown. We have experiments, labeling experiments, where we can track the path of a carbon into the soil and figure out which species of microbes are taking up this carbon. Now, we are very excited about these results. We're hoping to go into the field this summer to do large-scale experiments, which is always very important before any strong conclusions can be made. we would like to identify microbes that aggregate the carbon into stable soil organic carbon pools and that keep those carbon stable aggregates underground. So, and we need new tools to quantify how long this carbon is underground. I mentioned trials must be scaled to assess reproducibility across diverse soils and cropping systems. So obviously we're not ready to hand out these seeds yet. And I just want to end with saying that we and others are very excited about using agricultural systems to both draw down carbon dioxide into the soil and reduce methane emissions. But this is a long-term process. It's still essential to reduce fossil fuel emissions and develop other ways to pull carbon dioxide down. So we need all these tools as we move forward. So thank you very much. Thanks for listening. I look forward to the discussion.
Thank you very much, Professor Christine Pamela Ronald. Thank you so much for your informative speech. Ladies and gentlemen, and now it's time to move to the second keynote lecture. Let us welcome Professor Raphaël Mercier, Director of Department of Chromosome Biology at the Max Planck Institute for Plant Breeding Research, Germany. Please welcome Professor with the presentation on sexless seeds for better crops. So Professor, the stage is yours.
It's my great honor to be here and to present an innovation that can contribute to face the problem that was discussed before. Before I go into the topic, I'd just like to say that what I'm going to present is the result of a very exciting internal collaboration with my colleagues which are in this corner here. So I'm on stage now, but this is our shared work. So you can also talk to them if you have questions after that. So I'm going to talk about sexless seeds. And to let you understand what we're talking about, let me get back to the basics. So sexual reproduction is the way plants, animals, and other eukaryotes reproduce mostly. So you are the product of sexual reproduction. Plants you eat are the product of sexual reproduction. So So you start from an organism which has typically 2 copies of each chromosome, and then this organism produces gametes, which then has only 1 copy of each chromosome. And the next stage is two gametes to fuse to get back to an organism with 2 chromosomes. The 2 key events, really central events in this process, which again conserve between different eukaryotes, is meiosis, a very specific cell division that produces at the end the gametes. And I would advocate that this is the most important cell division, most important biological event in life, maybe because I've been working on this topic for the last 25 years. So during meiosis, chromosomes are shuffled in addition to be halved. So at the end of meiosis, you have chromosomes like this, which are a combination of the previous chromosomes. So your genome was designed in the meiosis of your mother and the meiosis of your father. So the first moment you started to exist is when those chromosomes were produced. And this is also true for the plants. And the next event, which is very important for this sexual cycle, is fertilization, when those 2 gametes with recombined chromosome fuse to produce the next generation organism. Okay, that's the basics. Some plants are able to bypass or to tinker with this mechanism and can produce seeds without sexual reproduction. I call that sexless clonal reproduction through seeds, or apomixis. Apomixis is a botanical word for it. And this is not extremely common, but still many species can do that. Maybe one which is iconic is, I will learn at the end how to use this device. This Taraxacum, the common dandelion. The common dandelion mostly reproduces through apomixis through clonal seeds. So when you blow these things, I'm sure you have done that one day in your life, you propagate clonal seeds. And this mechanism, clonal seeds, could be very useful for agriculture. Why is that? You may know that first-generation hybrids are typically more vigorous, more robust than the two parents. So this has been observed more than a century ago and has been heavily used in agriculture. And many crops in our fields are F1 hybrids because of this gain, which is almost for free. It's the genetics of the plants that give this vigor and this increased yield in particular. But this is true only for the first generation. If you take the seeds from this F1 hybrid, because of sexual reproduction, because of meiosis, then the next generation is much more diverse and has lost mostly the benefits of the first generation hybrid vigor. If we could introduce in this F1 hybrid apomixis, clonal reproduction through seeds, then you will get, at the next generation, plants which are genetically identical to the F1 hybrids. So they will become like immortal hybrids of the first generation forever. You can propagate them for as many generations as you want. And this really changed the game for several reasons. First, it will be much easier to use these F1 hybrids because you can just harvest the seeds. You don't need to control the crosses here, which is a very demanding in terms of work and money at the end to produce these F1 hybrids by controlling the cross. So you could make that in a much easier way and then make seeds available for more farmers. And also this could be used in more crops where controlling the crosses is very difficult. And finally, in the current scheme, you need to control the two parents and to keep the two parents to produce F1 hybrids. If with this system you don't need the parents anymore because you produce a hybrid, you just need the hybrid to propagate it. So it also will change how hybrid breeding can be performed. Okay, how do we do that? How can we trigger this clonal reproduction through seeds in crops? Because I've told you that a certain number of plant species, of wild species, are able to reproduce by apomixis, but none of our crops are able to reproduce by apomixis. So what we design is a synthetic way to trigger sexless seeds in crops. So how do we do that? We need to modify the two key events of sexual reproduction. So the first one is meiosis, and we need to turn this meiosis division, cell division, into another kind of cell division, which is called mitosis, which is a much simpler division that simply produces two cells which are identical to the initial cell, in contrast to meiosis that both reduce the number of chromosomes and shuffle the chromosomes. And accumulating knowledge by basic research, we identified a way by turning off 3 genes to turn meiosis into a mitosis. First in a model plant, Arabidopsis, and then in collaboration with my colleagues in rice. But this is only half of the equation. The second half is fertilization. You need to trigger embryogenesis in the gametes you obtain from this mitotic-like production of gametes. So with this step here, you have now gametes, female gametes, which have all the genetic information of the mother. And if you trigger now embryogenesis without fertilization, you will get the next generation clonal individuals. Okay? So we modify both of these key aspects, one by disrupting 3 genes, and the other one by changing the expression of a specific gene. And okay, this sounds like science fiction, but it's not. So I'm getting close to my last slide. This picture is from this collaborative paper here. Here what you see is a F1 hybrid of rice, and the plants you obtain as the next generation by sexual reproduction. You can see here you have some that are much shorter, they are very diverse, and they don't have the quality of the F1 hybrids. Here, you have the same thing. You have the F1 hybrid here, and the progeny, the next generation, obtained by synthetic apomixis, by this mechanism we put together to get clonal seeds. And you see that the hybrid vigor and the plants just look exactly the same because they are genetically identical. So it works. We can propagate the quality of the F1 hybrids by this synthetic apomixis. Now we just need to harvest the seeds on this plant to get the next generation identical. So, in summary, synthetic apomixis can increase the availability of hybrid seeds, and then we can use the benefit of hybrid vigor in more varieties. So we can develop more varieties for different kind of agriculture, for different kind of uses. We can also apply this in more crop species in the future. I have in mind, for example, wheat, in which we don't use apomixis, we don't use hybrid vigor because it's too complex. But in the future, we can dream of having apomixis and then hybrid vigor harvested in wheat. And also, because now farmers can reproduce the seeds themselves, we hope we can give access to high-quality seeds to smallholder farmers. And the general theme of today is to say that we need to combine all the solutions that we have in front of us, to combine different expertise, soil science, genetics, anything you can think of to build together the future of agriculture. And with this, I thank you for your attention.
Thank you so much, Professor Raphaël Mercier, for your insightful speech and presentation. Thank you so much for coming with us. Distinguished guests, ladies and gentlemen, and now we are honored to welcome to the stage Dr. Nadia Radsman from Sainsbury Laboratory, Cambridge University, United Kingdom. Please welcome Dr. Nadia Radsman to deliver the lecture of Beans and Tiny Bioswitches for Climate-Resilient Agriculture.
Thank you. Right, so today I'll be talking about two things that I'm very passionate about, which are beans and bioswitches. Before I start, I would like to emphasize the importance of nitrogen in our food system. So nitrogen is the building block for proteins, and it is first incorporated into amino acids. If we think of how nitrogen first enters our food system, from the soil, it's actually through plant roots. And we get the nitrogen and the proteins by eating the plants or eating the animals that had eaten the plants. Nitrogen is also critical for plant growth and productivity. So during the Green Revolution in the 1960s, we know that the use of nitrogen fertilizers increased our global crop production. However, nitrogen fertilizers usage and production of this fertilizer is very detrimental to our environment. This is because the production process, the Haber-Bosch process, requires high amount of fossil fuels and also releases high amount of greenhouse gases. So more sustainable alternative is through biological nitrogen fixation. And this can be carried out by special bacteria in the soil. These bacteria have nitrogenase enzyme, and this enzyme can fix nitrogen from the air and produce a form of nitrogen that plants can take up. The ones that I'm very interested and also studying are symbiotic nitrogen fixing bacteria that can associate with a group of plants called legumes. So legumes are plants from the Fabaceae family, or the bean family. So think of soybean, lentils, chickpea. Those are all legumes. And what's interesting about legumes is that they can form these structures on the root system. So these structures here are called nitrogen-fixing nodules, and they house the bacteria inside these structures. The bacteria can then fix nitrogen from the air and provide plants with pretty much free nitrogen fertilizers. So legumes are reliable sources for sustainable nitrogen and sustainable protein. And in a way, we should be growing more legumes so that we can reduce our usage of nitrogen fertilizers. But here comes the tricky part. Legumes or bean crops are very, very challenging to genetically improve for climate resilient traits. So how do we solve this? One way is to use peptides as tiny bioswitches that can turn on and off particular biological pathways that can regulate plant growth and development. So examples of peptides, even though they are from animals, are actually insulin and GLP-1. So peptides are made from amino acids. So if we can think of one amino acid as one bead, peptides are usually less than 100 beads long. And they are way smaller than proteins. So proteins are usually thousands and thousands beads long. In a way, peptide can be seen as very, very tiny proteins. So to demonstrate how peptides can be used as bioswitches, I'll be presenting 3 different peptides that I've worked on before and currently still working on. So the first peptide is the CEP peptide, or C-terminally-encoded peptide. And this peptide can increase nitrogen-fixing nodule formation on legumes. So in legumes, high nitrogen can inhibit the formation of these nodules. So we can see here, we can see here at 5 millimolar and 25 millimolar nitrate, there is reduction in the formation of the nodules. However, we can bypass this inhibition by adding the peptide, and we can increase the formation of the nitrogen-fixing nodules. How this works is that the CEP peptide is actually a nitrogen limitation signal that the plant roots actually produce. The peptide is then transported into the shoot to actually signal the shoot that, hey, there is actually low nitrogen in the soil. And in response, the shoot would actually produce another signal down to the root, and this would change the physiological response of the root. In this case, it's to increase the nitrogen-fixing nodule formation. The second peptide is a collection of peptides that can accelerate plant regeneration. So as we've seen from previous talks from Professor Mercier and Professor Ronald, we need to improve our crops by gene editing. And part of editing genomes in plants requires tissue culture process. So it involves having a particular plant tissue and inducing callus formation. So callus is pretty much equivalent to stem cells. After that, we need to induce the formation of the shoot and the root in order for us to actually get an improved gene-edited plant. For legumes, this particular process here is very, very challenging. A lot of legume species cannot make the shoot or the root after callus formation. And because of that, it's very difficult to actually get an improved gene-edited legume plant. To improve this, what we can do is actually use peptides that can accelerate and force the callus to actually make the shoot regeneration or root generation. So this is an example of one of the peptides that I'm studying. So we can see here, this is shoot regeneration on the model plant Arabidopsis, and in the presence of the peptide, you can see that there's accelerated shoot development, and there's even a flower bud here after 2 weeks of induction. The third peptide is E040 peptide, or early nodulin 40. And this particular peptide is expressed in the vascular tissue of nodule. So if we cut the nodule here, what we can see is that it is expressed in the vascular tissue. So vascular tissue is responsible for transporting carbon in the form of sugar in plants. This particular peptide is encoded by this very strange gene. So it is encoded here at the 5' end. And we know what the peptide is doing to the plant. It increases the activity of a particular enzyme responsible for carbon resource allocation. So carbon resource allocation is important, for example, if we want to increase fruit formation or grain formation, then allocating more carbon to the shoot would actually improve this. And 40 can accelerate this process. What we did not know for the past 30 years is actually what this red region is doing. And recently, we found that it's actually a mimic of a microRNA. So microRNA, we can think of it's an inhibitor of a process. In this case, it's inhibiting the carbon resource allocation in nodules. So by having this particular red box, red region here, what it's actually doing is it is inhibiting the inhibitor. And because of this, it's actually reinforcing the positive regulation of 40, so then we can have better carbon resource allocation for nitrogen fixing nodule formation. Right, so to summarize my talk, legumes are important for sustainable agriculture, especially because of their nitrogen fixing capability. And we can improve legumes by using peptide bioswitches that can increase nitrogen fixation, can accelerate plant regeneration, so we can have better crops, and also improving carbon regulation. And thank you for your attention.
Thank you very much, Dr. Nadia Razman, for your presentation. Thank you so much. Distinguished guests, ladies and gentlemen, and now it's time for us to move to the last keynote lecture. Please welcome to the stage Professor Ermias Kebreab, Associate Dean for Global Engagement in the College of Agricultural and Environmental Science, and also the Director of the World Food Center at the University of California, Davis, USA, with the lecture on Scaling Climate-Smart Livestock Innovations for Productivity and Security in the Global South. Welcome, professors. Thank you for coming with us, and the stage is yours.
Okay, well, thank you very much for the introduction, and thank you for the opportunity to come here and share this with you. So what I'm going to do is I'm going to take you away from the world of plants and introduce you to the world of livestock and animals. As we all know, livestock have a lot of importance. They are vital for our livelihoods, for providing nutrition, and they are extremely important for rural economies as well. However, on the other side, we have a problem of generating greenhouse gas emissions as well. So from livestock, about 14.5% of the global greenhouse gas emissions is coming from livestock. But we also have a way to reduce these emissions as well. Particularly in the Global South, we have quite a lot of different tools that can be deployed at scale that we can use to reduce those emissions. So ideally, what I would like to see is the triple win, where we increase the productivity, because there is quite a big yield gap that's happening in terms of livestock productivity. So increase the productivity and increase the circularity. So basically what we mean by circularity is use waste products, byproducts, I should say, and use those for animal feed so that we can increase productivity that way, so we have the circularity of the whole system, and also reduce the greenhouse gas emissions, particularly methane emissions as well. So we can achieve all these three, that will be really great, and there are ways to do that. So I'm going to sort of give you examples from Vietnam. In California, what we've done the last decade or more is really looking into byproducts and integrating them with animal production. One success story in California, for example, is the almond hulls. Almond hulls used to be sent to a landfill. California produces a lot of almond hulls. In fact, it's the only state that produces almond hulls in the US. And so a lot of this will be just sent out. But now, if you visit any dairy in California, you will see mountains of almond hulls. That's come from research saying that about 8%, 10% of the feed could be almond hulls, and animals produce really good amount of milk. The same thing with tomato. Again, if you go to California, you see trucks and trucks loads of tomatoes, and after you make the ketchup, there's a lot of stuff left over, and that is now used for animal as well. And at UC Davis, recently we've done work in grape pomace. So again, California produces one of the best wines in the world. I'm saying in front of French people here, but, but there's a lot of grape that's left after you make the wine. So what happens to that? Now, we've done some work at UC Davis where we show that if you use that, you can increase productivity and reduce greenhouse gas emissions as well. So similarly, we're trying to work here in Vietnam with Vietnam National University of Agriculture. We're using locally available products, like the Vietnamese jungle tea. It has some extracts that will help reduce the use of antibiotics in animals, and then also improve the productivity of animals as well. So we're hoping the VinFuture Foundation would support these kind of projects. With Vin University, we're trying to look into aquaculture and work in terms of developing some technologies where you can look into monitoring technologies, so you can increase the productivity of those of industry. And we're working with Can Tho University, where using byproducts, again, they're all available here in Vietnam. Cassava leaves and pulp waste is highly available. Use that to feed animals, increase productivity, as well as improve the circularity of the whole system as well. And just give you, again, a bit more detail on some of those technologies. One is Gracilaria and Asparagopsis can be used as feed for Vietnam. Here you see farmers, this is actually in India, but this could just as well be in Vietnam, where farmers are out there collecting the seaweed, and that seaweed can be used and processed as a fertilizer and can be used for high-value crops. And then there's a lot of that seaweed left, a lot of biomass left, and that biomass can be integrated with other byproducts, especially for fish processing, where it has high levels of protein. You can put that together and becomes a really valuable animal feed as well. Using Asparagopsis, you can actually reduce greenhouse gas emissions, particularly methane emissions, quite substantially. By up to 90% of methane, enteric methane, can be reduced by using some species of Asparagopsis. So you have a lot of benefits. You reduce the pollution, the nutrient loading of coastal waters. You have a lot of nitrogen and phosphorus and stuff in the coastal waters. You can take all of that through the seaweed, use that for crop production, and the rest you can use it for animal production as well. This is the work that we've done in the US. We could just as well do it here. In fact, those two machines you see here, they are state-of-the-art sort of devices to measure emissions. And we have those now installed at Long Dam University in Ho Chi Minh. And if you go today, you will see that there are animals that their emissions have been measured. So this is the first time, actually, breeds from Vietnam, we are measuring their emissions right away. And so what we see here is we give them some seaweed, particularly Asparagopsis, and we've seen is, I got too far. All right, so here you will see that as long as they consume the seaweed, which we call the brominata, there's a sharp drop in methane emissions. So up to 40% in this case in general. But we've seen some animals, they have 90–95% reduction, and other animals about 10%. It depends on how much they are eating. The more they eat, the more reduction that we're going to get in this way. So we are introducing this in Vietnam as well. For this project that we are doing, we're using oil byproducts, because these are some of the products that are highly available in Vietnam at the moment, but then we are going to move into seaweed and others that are also can easily be grown in Vietnam. And the other aspect of this is precision feeding. So understanding what the animal actually needs and then feed them exactly what they require in order to produce a certain amount of milk or meat. One of the biggest challenges is that to figure out what is the nutrient quality of any ingredient that you have. So in the US, what we do is we collect samples, we send it to the lab, and then in a couple of days, you get the whole thing, but it costs a lot. For each one, you probably cost about $30, $40 for each ingredient. So it's not really reasonable for smallholder farmers to do that. So we're trying to put together, and this is the near-infrared system where we can calibrate it so that we can get the quality of the feed right away on the field. And from there on, we are actually developing something using just your mobile phone. You take a picture, and that picture, through AI and machine learning, will translate into the amount of carbohydrates and protein and lipids and minerals and all that kind of stuff. So this is in development right now. And at the same time, we are developing a nutrient requirement for tropical cattle. So we need to know what they require. We need to know how much we have. And then we put all that together into a tool, into a ration formulation tool. And then this will then give you the recommendations of how much of each of the ingredients you should be consuming. And what we're trying to do is at the end, something like, I'm sure everybody used ChatGPT, similar like that will be what we call the farmer chat. And you don't even have to type it in. You can just say into your mobile device, saying, I have a cow that's producing 5 liters today. I want that cow to produce 10 liters. And I have available for me elephant grass and this concentrate and that. And then, or you take a picture, and all of that will be integrated, and then it will give you the recommendations in the language that you use to ask the questions as well. So this is in development, and we hope they will be finalized. The initial version of this was released back in October actually in Kante as well. So just to summarize, there are the different ways in which we can achieve reduction in emissions, improve productivity, and have the circularity using the byproducts that are available locally. So if you can do this, then we have the triple win that I talked about earlier: higher productivity, better circularity, utilization of resources, and methane mitigation as well. Thank you.
Thank you very much, Professor Ermias Kebreab, for your presentation. Thank you very much. So, we've been through 4 keynote lectures from our distinguished guests. And before moving to the panel discussion today, may I invite all of you to be with us to follow a short video that is the recap of the VinFuture Prize Award Ceremony 2024. More to follow.
Entering its 4th year, the 2024 VinFuture Prize Award ceremony has firmly established itself as one of the most anticipated annual events in the global science and technology community. This year's award ceremony was held in the magnificent Ho Guam Opera House, graced by the presence of esteemed Vietnamese government leaders led by the Prime Minister Pha Minh Chin, along with world-leading scientists and technologists whose groundbreaking innovations have empowered humanity's resilient rebound, shaping a sustainable and prosperous future. For the 2024 VinFuture Prize, a record-breaking 1,469 nominations were submitted from over 80 countries and territories, showcasing the global appeal and significance of this prestigious award. Let us applaud the groundbreaking technological solution meticulously chosen for the VinFuture Prize. The VinFuture Special Prize for Innovators from Developing Countries was awarded to the Bangladeshi scientist Dr. Firdausi Kadri for her key role in improving a single oral dose of affordable vaccine against cholera, a severe diarrheal disease which remains a major public health concern, particularly in areas with poor sanitation and limited access to clean water. This groundbreaking innovation subsequently controlled cholera outbreaks at the source, enhancing global public health security. Acknowledging the significant contributions of female scientists, The VinFuture Special Prize for Women Innovators honored Professor Christy Anseth for her pioneering advancement in the design of polymeric biomaterials and methods for biomedical applications. Her field-defining experiments elucidate the role of extracellular matrix signaling on cell fate, which is foundational for studies and insights into tissue maintenance and regeneration, laying a path for the development of biomaterials for regenerative medicine. The VinFuture Special Prize for Innovators with Outstanding Achievements in Emerging Fields was given to Professor Zelig Eshar, Professor Carl June, and Professor Michel Sadelaine for the revolutionary development of CAR T-cell therapy for cancer and other diseases. While Professor Zelig Eshar's groundbreaking work transformed cancer treatment by developing CAR T-cell therapy, Professors Carl June and Michel Sadelaine built on this progress, further improving CAR T-cell therapy to effectively treat cancer and autoimmune diseases that resist standard therapies. And finally, the prestigious $3 million VinFuture Grand Prize was awarded to Professor Yoshua Bengio, Professor Jeffrey Hinton, Mr. Jensen Huang, Professor Yan Lukun, and Professor Fei Li for the transformational contributions to the advancement of deep learning, comprehensively acknowledging all three aspects of algorithms, data, and hardware. Professors Jeffrey Hinton, Yan LeCun, and Yoshua Bengio have made groundbreaking contributions to neural networks and deep learning algorithms, while Mr. Jensen Huang's graphics processing units, GPU innovations, significantly accelerated the efficiency of computations. Professor Fei Li's creation of the ImageNet dataset further drove advances in visual recognition systems, making it possible to train models at scale. Experience a cultural fusion like no other with the electrifying atmosphere as the iconic rock band Imagine Dragons graced the stage with the explosive performances. The audience was also mesmerized by the passionate violinist Hoang Ho Can Van, the talented MQ dance team, and the members of Vietnam National Opera and Ballet. Let's celebrate the 2024 VinFuture Prize spirit, Science for Humanity's Resilient Rebound.
Thank you so much for your attention to our recap video of VinFuture Prize Award Ceremony 2024. And don't forget to be with us. Follow the VinFuture Prize Award Ceremony 2025. Will be live broadcast in VTV1 December the 5th. And we will find out who will be the laureate of the special prize, and especially the grand prize of this year. Thank you so much, ladies and gentlemen. And now it's time for us to come to the symposium's today's discussion. May I introduce to you and welcome back to the stage Professor Ingolf Steffan-Dewenter, the chair of today's symposium. Welcome back to the stage Professor Pamela Christine Ronald. Welcome back Professor Raphaël Mercier, Dr. Nadia Razman. And last but not least, welcome back to the stage Professor Ermias Kebreab. And this is our panelists today. And the symposium discussion will be moderated by Professor Ingolf Steffan-Dewenter, and the stage is yours.
Yes, thank you very much. A warm thank you to our panelist speakers and giving these exciting presentations on solution for future more sustainable agricultural systems. So as we acknowledged, there are major pressures on agriculture, there are major challenges for food security, so we need a wealth of innovations to promote the sustainable of food production systems in the future. And we heard here about very promising and different solutions for future agriculture. So I would like to start first with livestock innovations. And Ermias, you presented that there's a huge benefit of feeding seaweed to livestock in Vietnam, so reducing methane emissions by 90%. And so I wonder, is that a general pattern? So is there a trade-off between the food quality and feeding certain food components that reduce methane emissions, or is it a synergy? Can you do both, having a circular waste economy, reducing methane emissions by feeding these seaweeds in your case study, and having a high production level of the livestock system?
Great, thank you for the question. So there are a couple of different types of seaweed. So if you use the Asparagopsis, a red seaweed, and one particular species, Asparagopsis taxiformis, and other seaweed within the same genus as well, those have particular metabolites that have the ability to basically reduce the methane, and they interfere with the last phase of methanogenesis, and that's why you reduce methane emissions. So, but that's just one particular seaweed. Luckily, that seaweed grows in Vietnam really well. So it requires a warm kind of water. So off the coast of Australia, Vietnam, those are places where you will see the production to really increase. Right now, it's not been produced, it's not mass produced because there's no demand yet, but it's still at the research phase. But that's what we're hoping to see, is that this production continue with that, and you don't need to include a lot. Just a few grams will do the trick, and you will have a 40, at about 28 grams of bromoform per kilogram, you will get about 40% reduction, 50% reduction. If you increase it, you can go up to 90-95% reduction. But the other thing is, other common seaweeds that do not have the bromoform, they are also very useful, because those can also contain different amounts of proteins and lipids and other nutrients that animals require. And so they could be part of the diet for animals as well.
Okay, and one follow-up question, is this a feasible solution for other regions, also not only in the global south, but also in the global north, with high-intensive livestock systems?
Yeah, absolutely. So the Asparagopsis is actually been developed a lot in the global north, because you need a lot of inputs and research that need to be done. So that's a lot of the research has been done in the global north at the moment. But certain areas like Vietnam, because of the ability to produce those, will become relevant. But in other global south, that probably is not the first thing that you want to do. The first thing is really improve productivity, because there's such a big gap in productivity. Cows, genetically, you could get maybe 12,000, 13,000 liters of milk per year per cow, and some people think by 2050 you can get up to 20,000. And in most low and middle income countries, it's about 2,000 and less. So there's a huge yield gap that we need to close before we think about just reducing methane emissions.
Okay, thank you. So I may turn to Nadia. So your research is very promising, because, as you mentioned, external fertilizers are one of the major pressures on the environment. On the other hand, they are mandatory to maintain crop productivity in many systems. So I wonder your approach to use these small peptides to increase the nitrogen fixation in legumes. How close is it to application in crops, and could it be used not only for legumes but also for other cropping systems? So what is your perspective on this?
Yeah, so in general, for example, the SAP peptide that I mentioned in my talk, it can also increase nitrogen use efficiency in non-legume crops, and this is by increasing the nitrate transporters. So we can actually use it not for making nodules on non-legume crops, but actually increasing nitrogen use efficiency. However, having said that, as you mentioned, how far is it before we actually can use it in the field? The problem with using peptides at scale is the production cost. And currently, there is no reliable way to pretty much spray the peptides at scale at cost-effective rate. So there should be bridging technologies that can allow that, in order for us to use all these different peptides that we have discovered including the ones that Pamela already mentioned in her talk, the Cy1. There's also a peptide, and there's also a potential that rather than using genetic transformation to overexpress it, we can should be able to just spray them. But again, we need to have the bridging technology. Yeah.
Yeah. Okay, thank you. So it sounds like a very promising pathway. So there already now there are mixed cropping systems that combine legumes with other crops to make use of the nitrogen fixation capability of these legumes. So imagine that they can enhance this nitrogen fixation. It could be a game changer also in mixed cropping systems. Yeah, thank you very much. I would like to turn to Pamela, and so you are an expert in plant engineering, and you have shown these exciting developments to increase not only drought resistance, but also the level of methane emissions of rice in one of your key examples. So each example, it sounds extremely promising, and I wonder, would it be possible to combine different genetic engineering approaches to have a crop that is drought resistant and perhaps have higher nitrogen fixation capabilities and is emitting lower methane to the atmosphere. So is that a plausible perspective, or is it beyond the current technology?
No, absolutely. That's entirely possible for all scientists. The key often is the fundamental research, to make the fundamental discoveries, to understand, how the peptides are affecting the metabolism. And if we're lucky and we fall on a gene that has a broad effect, so for example, the sub1 gene, I think that you probably know about, confers submergence tolerance, and it's been very useful. 10 million farmers are growing it now. And people at the International Rice Research Institute, breeders are crossing it to all kinds of different lines that are resistant. And so this is something fundamental that geneticists have been doing a long time, is making crosses, and we can call it stacking genes. So you stack all the good genes in the best variety. So whether it's a transgene or a gene introgressed or introduced through marker-assisted breeding, all of those can be stacked. And I think that's what Raphaël was alluding to, putting some of these self-cloning crops, if you can also make them submergence tolerance, you can have them maximize their nitrogen fixation, you can figure out what microbes in the soil are going to affect methane emissions. There is no reason they can't be stacked together.
Okay, yeah. So perhaps a follow-up question. So you have written a book on organic farming, plant genetics, and I think the future of food. So how could this come together? So organic farming, at least in the German context, is the opposite to plant engineering. So how would you combine these very different technologies, plant growing systems and then plant engineering?
So my husband's an organic farmer, and I'm a geneticist, so sometimes we'd go to parties and people would be like, how can you even talk to each other? And we have the same vision, which is to advance sustainable agriculture. So we're not ideologically biased, and we just, in the book, we go through many different discoveries, genetic discoveries, applications, and we think about certain criteria, whether they can be broadly useful or accessible to many different populations, whether they are measuring the benefits versus potential consequences that one can't foresee. So we sort of go through each of these, and the bottom line is organic farmers love seeds. So Raul has a chapter in the book where he's just sitting looking through his seed catalog, and that's what he does a lot of the time now, too, I think, when he's not kayaking. And so the seed is just so important to every agricultural system. Organic farming is very focused on farming practices. The reason that you cannot put them together is because of the regulatory environment for the National Organic Program Standards in the United States specifies you cannot use genetically engineered seed. So we're not trying to change regulations. That's not really, we're not so good at that. But we thought that, well, maybe we could look at the other 98% of agriculture, because in the US, organic farms is maybe about 2% of agriculture. So how can we make the other 98% of agriculture more sustainable? And we found that the answer was to incorporate some organic farming practices and incorporate some of the best genetically engineered seed.
Yeah, thank you very much. I fully agree. And I'm also a passionate gardener and love to look for seeds and different seed varieties. So, but that's also a great keyword to turn to Raphaël. And so you have this fantastic innovation to develop apomictic seeds that don't depend on cross-fertilization anymore. And so my first question is how, so you had this one example with rice that is near to implementation level. Was it a very long way to come to this step? And is there a near future where we have a multitude of different crops and varieties that can be produced by this technology?
So the story behind rice, we started with Arabidopsis 15 years ago, and then took time to transfer to rice with my colleagues here to have the first part of apomixis. And then the lab of Sundaresan here identifies the second part. Then we put everything together in 2019. So it's not that long ago. And us and other labs also play with the system to improve the seed production of those hybrids. And now they are ready for the market, really. They are really in rice. So rice is really the flagship of this technology, which is now ready. Now we would like also to implement that in other species. So several labs, including ours, work on barley, tomato, soybean, and on paper, there is no reason we cannot manage to do that. We just need to adjust the system. So it will take some years, but nothing, I think, is really blocking the innovation to be transmitted to other crops.
Okay, so that sounds like a quite rapid next step that is possible.
Maybe as a follow-up of the previous question, I mean, I would love to have this technology implemented in organic farming. That would be great. So for me, this technology should be used in any systems because we have the same goal, all of us. The regulation is a difficulty for organic farming, but also in Europe, we cannot use, at the moment, geomediting. And this is something which is really blocking innovation. So I hope that this will change soon, and we can implement the technology also in Europe and not also only in non-European countries.
Okay, yeah. So perhaps one follow-up question. So in many cases there are studies demonstrating that there's a regional adaptation also of crop varieties to the local soil conditions, to the climate conditions, also to the microbiome in soils. And this implies that we depend also for crops on constant adaptation, on microevolution. And I would like to know how this component could be included in the technology you have developed to make sure that the novel crop varieties or apomictic seeds are also adaptable to very different climatic conditions, for example.
Mm-hmm. So what you are pointing to is the importance of plant breeding in general to generate new varieties adapted to all the environment you described and all the needs you described, like adapted to less nitrogen use, for example, or anything we can think of. So this is plant breeding in general, which is very important. Apomixis or clonal reproduction through seed should be included in the plant breeding. And that's only the final step. The idea is to create varieties, hybrid varieties, with regular breeding, and then to turn on apomixis to propagate this line that you produced. There is one image which is used sometimes in the field which I like, which is the invention of the printing machine. So the printing machine is a cloning system. And because this machine was invented, we had an explosion of the diversity of books, from just a few books which were just copied by hand to an enormous diversity of books that we see today. So inventing a cloning machine actually favors the diversity and not the opposite.
Okay, thank you very much. So I assume we have a little bit more time. So I would like to have a quick round of answers, just a brief answer, please. But from your perspective, what are the main or the most promising combinations of future innovations in agriculture for mitigating environmental impacts, for ensuring food security, but also what are the main barriers to achieve these future innovations? So we just start in the same round as before, and not too long, please.
Yeah, sure. I mean, in terms of livestock, I think the way that we need to move forward is really in increasing the productivity, because increasing productivity actually gets you a reduction in emissions per unit of products, right? For a kilogram of milk or per kilogram of meat, you will have less emissions if you increase the productivity. At the same time, that would be that's something that the farmers would want as well. And so you have an economic benefit, you have an environmental benefit as well, and you're meeting the demand as well, because the demand for animal products is increasing. We talk a lot about alternatives. That's great. I think we should have different alternatives, but the reality is that the demand for animal source foods is on the rise, and it will continue to increase. So what do we need to do? Do we want to have low-producing animals, just increasing the number of animals, or having less animals but more productive animals? So I think that's where the focus I see in the future.
Okay, thank you. Nadia.
Right. So for me, I think the innovation that I'm excited about is the gene stacking, as you mentioned, through, I would say, very smart decision-making using system biology. And I think the other answer on the challenges is actually democratizing plant biotechnology to Global South.
Well, what we really need is more funding for research. Because
you always agree.
The fundamental knowledge is so important. These exciting discoveries we're hearing about has taken years. I remember when I first came into the rice world, maybe 35 years ago, the dream was apomixis, and it was just about to happen. And it's taken a wonderful team of scientists working in different areas to bring this forward. So I'm very excited about this, and it's really a global collaboration too. Politically, we need to continue to support global collaborations, and of course everything we've talked about here I think is very important. One thing that we didn't mention is, of course, AI is becoming more and more important. Everybody's using it. Our students now are creating models that are helping to predict the function of genes that we never knew before. So that absolutely is accelerating our research.
Okay, yes, thank you.
We need to combine everything, but we need our decisions to be based on science, on facts. So we need to combine AI, soil science, genome engineering, all of this together and do even by science. So we need to break the dogmas, to break the barriers between fields. That's what we are trying to do today. And what I wanted to say, and yes, and we need research. So we need to fund basic research and applied research to reach this. And funding basic research is really important.
Thank you so much for your conclusion from our panelists. So, ladies and gentlemen, one more time, please give a warm round of applause for the discussion from our distinguished guests and panelists and the chair. And now it's time for me to help the panelists to open the Q&A session with the questions from our audience. So if you have any questions, please raise your hand so I can assist you with the microphone over. Yes, please, from the front row. Yes, our staff can assist with the microphone.
I have a general question. You didn't talk, or was not the topic of this panel, to talk about the waste of food. I heard that a lot of food is wasted, about 30%. Not heard, I mean, I read in many places the FAO mentions that about 30% of the food is wasted. So we talk about increasing productivity and so on, and that's fine, of course, but if we waste 30% of what we produce, I mean, my eyes is something strange, no? Produce more and waste more and more. So just a comment about this issue of the waste of food.
Yeah, I might answer to this because I raised this point. I also feel it's a very important aspect. So we always talk about food security and the need to increase food production by 100%. On the other hand, we have a lot of food waste after the harvest. And in addition, we have this yield gap before the harvest. And we have the potential, as we have here today, to increase the yields, so to have perhaps an even larger yield gap depending on how other ecosystem services work. So that means that we need to find solutions that aim for reducing food waste, to increase productivity and sustainability on existing agricultural land, and to reduce the expansion of agricultural land to the remaining natural ecosystems.
Thank you so much.
I can just quickly add to that.
Sure.
In low and middle income countries, the waste, it's about the same. In high income countries, about what, 30 to 40% is wasted. Low middle income countries, 30 to 40% is wasted. But the difference is, in high income countries, the waste is after the food has been harvested, often when it's on your plate. In low and middle income countries, the waste happens before it gets to the plate. So that's a really key area where scientists have been spending a lot of efforts to develop plants that are resistant to pests, resistant to pathogens, resistant to climate change, for example, drought, heat, submergence. So that's something that we can do as scientists, molecular biologists, look at the waste there. But I think the waste that many of us in high-income countries, that's a societal, cultural issue.
Yeah. It could be changed.
Yes, it has to be changed. Right.
Thank you, Professor Pamela, and thank you, Professor Ingolf, and thank you so much for your questions. And we still have the room for another question, so could you please raise your hand so I can assist you? Yes, please, Professor Pascal Cosset.
Thank you very much. That was really interesting. I have a question which makes a link with what we heard this morning. So you said that, this is a question for Ermias. You said that the Vietnamese jungle tea can help reduce use of antibiotics in animals. So the use of antibiotics in animals has been prohibited in Europe. And for the people who are not aware, the use of antibiotics in animals is to help the body mass to increase. So people are giving antibiotics to animals to increase the speed of having body mass. I think it's fascinating to say that the Vietnamese tea leaves could be used. So do you know what is the effect on the microbiota? Do you know if it is really used, or can you comment on that?
Yeah, sure. I mean, I definitely agree with you in terms of antibiotics. The antibiotic resistance, particularly in low and middle income countries, is increasing because there's a lot of use of antibiotics. So we need to find alternative ways to do that. I mean, a lot of antibiotics is used as a growth promotant and all that, and used in a subtherapeutic doses, which that's what the problem is. With locally based ingredients, that's the research that needs to be done. So we need to do the research. Vietnam National University of Agriculture need to do the research to find out what are the active ingredients in those local plants that can be used as a replacement for antibiotics.
They are really effective?
Well, we need to do the research. So there's, right now, we need to do the research to find out exactly how effective they are. They are effective in vitro. So some in vitro work in the lab shows some activity to reduce infections and all that. But it has to be done in vivo, in using animals to
So
make
what is
sure
in
that you do
vitro?
that.
So what is in vitro?
Sorry?
What do you mean by in vitro?
In vitro means in the lab. You're just using, you know, you have,
Leaves and bacteria?
you have pure culture of microbes, and then you add the extracts and see if there is an antibiotic effect. So that's just in vitro. But a lot of the times, what it works in vitro might not be translated in vivo. So we have to actually work with animals in vivo to make sure that what we see in vitro is translated in vivo as well.
I thought you had given that already to cows or something. Not yet.
Not yet.
Okay.
We hope.
Thank you so much, Professor Pascal Cosset, for your questions, and thank you so much, Professor Ermias Kebreab. Yes, ladies and gentlemen, we still have time for maybe 2 more questions for our panelists. Yes, please, lady in red in 3rd row. Thank you. Yes, please.
Thank you so much. I'm from the Vietnam Farm and Agricultural Enterprise Association. So I'm very interested in about the Dr. Ermias' presentation. About the low emission in livestock, but I have 2 questions. First one about how to use the seaweed in Vietnam, because in Vietnam also we have a very long coastal area. So how to use the seaweed in the own season, or that's only for summer or winter, something like that. The second one, the second question is that in the Vietnam now, you also have a very high population in cattle and dairy cattle. In Vietnam now, we also have about more than 8.6 million cattle and dairy cattle population. But almost have some cattle in the mountain area. So in the mountain area, how we can use another herb instead of the seaweed? Is the second question for Dr. Ermias. Thank you so much.
Okay, so I think maybe I'll start with the second question. So there are different solutions for different situations, right? So the animals that are far away from the sea, the seaweed, it may not be the right choice to do that, right? So there would be other byproducts that are more readily available in those areas, so it makes sense to use what's available in that area. So that's why the solutions have to be really related to that specific area. We cannot have just one solution we try to fit into everything. It has to be really tailored to that region, to that specific area, and what's available in that area. But in terms of the production of seaweed, yeah, I mean, Vietnam has a long coast, and so there's a potential to produce this in large scale. And at the moment, the research is that it shows that it's very effective. There's no doubt about that. There's quite a lot of papers already. In fact, we even did a meta-analysis of all the papers, and we've shown that it's very effective. But it still needs to go through a regulatory approval process. There is a little bit of worry about the active ingredient, the bromoform, in terms of the doses, how much of that should be given. So once it's shown that it's effective for animals, it's okay in terms of health of animals, health for people of the products, and health of the environment, then there is a potential to set up a facility to produce this seaweed in mass and be able to even export it to other places. So those kind of things are happening on the coast of Mexico, US, and Australia. But I see that there will be a huge potential in Vietnam to do that once there is approval. Right now, there's no approval. It has not been approved anywhere in the world. There are some partial approvals in Mexico and Chile, but there is no full approval at the moment.
Thank you so much, Professor Ermias Kebreab, and thank you so much for all the questions from everyone in the audience. One more time, thank you so much for your attendance with active participation with our event today. And especially, I want to express my deepest gratitude to all of our panelists for your impressive presentation and especially discussion today. Thank you so much. And don't forget that we still have time for the breakout sessions that you and the audience and the scientists in the field can connect and network with each other in 3 different rooms, and we have 3 assigned topics as well, and the session will last 1 hour until 4 40 and 5. Thank you so much for coming with us, and we hope that you have a wonderful time with our symposium today. We wish you all the best with happy and happiness, and don't forget to follow the VinFuture Prize Award Ceremony 2025 live broadcast on VTV1 December the 5th. Goodbye for now and see you tomorrow. Thank you.